Hi, hello to all the students of BPC. This is your Prasanna ma'am from NEET World. So yes, we are here to discuss one more previous year question paper of CUET Biology from the chapter Sexual Reproduction of Flowering Plants, Principles of Inheritance, Molecular Biology, Microbes in Human Welfare and Biotechnology and its Applications. So if you look into the first question, firstly, why you have to solve this paper? This paper you have to look into because this plays a crucial role in attempting CUET further examinations like CUET 2025 if you are writing still, and CUET 2026 also. You will get to know all the patterns of the questionnaire that he is picking up from the concept. So it is very important to look into this session, ma. This is very useful for you. So yes, if you look into the first question, parthenogenesis does not occur in. Parthenogenesis is production of the offsprings without fertilization. Okay, that is parthenogenesis. Parthenogenesis is possible. Parthenogenesis is possible in these organisms like rotifers, honeybees, lizards. Parthenogenesis is possible. And in honeybees, what type of sexual determination is there? Haplodiploid type of sex determination. But whereas in mammals, parthenogenesis is not possible because of genetic imprinting of both male and female is required for the progeny. Okay, so parthenogenesis is not possible for mammals. Next question, if you look into, which of the following is not a Mendelian disorder? It is most important to note, beta. Mendelian disorders are of two types, autosomal and allosomal. Autosomal you have autosomal recessive, autosomal dominant. So yes, Mendelian disorders, you know, they are of two types. One is autosomal and the second is allosomal. Okay, autosomal and allosomal. Okay, autosomal we are again having two types, autosomal recessive and autosomal dominant. Allosomal are again of two types, that is X-linked and Y-linked. X-linked again we have X-linked recessive, X-linked dominant. Once let us quickly recall all the Mendelian disorders. Autosomal recessive disorders include phenylketonuria, sickle cell anemia, thalassemia, cystic fibrosis. All these diseases comes under autosomal recessive disorders. Autosomal dominant best example is myotonic dystrophy. When it comes to X-linked diseases, X-linked recessive diseases, most noted they are color blindness and hemophilia. Okay, color blindness and hemophilia. Up till here, this should be in your mind. When it comes to chromosomal disorders, we have learned three types of chromosomal disorders. One is Down syndrome, which is of trisomy of chromosome 21, which is of trisomy of chromosome 21. When it comes to chromosomal aberrations or chromosomal disorders in the allosome, Klinefelter syndrome, which is a trisomy in males, and Turner syndrome, which is monosomy in females. Okay, both of them are sterile. So of these four examples, if you see, phenylketonuria is a Mendelian disorder. Okay, autosomal recessive, hemophilia is X-linked recessive, sickle cell anemia is autosomal recessive Mendelian disorder. So yes, Down syndrome is not a Mendelian disorder because it is a chromosomal disorder. This happens because of trisomy in 21st chromosome. Next question, if you look into beta, this is a match the following type of question wherein polyembryony, that is many embryos inside the ovule, they are formed either due to different embryo formations because of nucellus and integuments, or due to embryo sac formation from the nucellus or integuments, which results in many embryo condition inside the ovule, which is commonly noted in Asteraceae and grass family. So yes, polyembryony is observed in orange. Okay, it is observed in orange amongst these examples. Parthenocarpy, that is seedless fruits. This is most common in banana, grapes. Okay, false fruit, wherein other floral parts along with the ovary are cooperating in fruit formation. So yes, false fruit example, best example if you take, that is apple beta. Next, embryo sac. Embryo sac is called as, it is the female gametophyte in the angiosperms. So embryo sac is the female gametophyte in the angiosperms. This embryo sac is haploid beta. So yes, if you look into the options, polyembryony, that is polyembryony, you will see it in oranges, that is option 3. Parthenocarpy, that is option 4. And next, false fruit is seen in apple. False fruit is seen in apple. Then embryo sac formation, that is embryo sac is haploid female gametophyte, that is option 2. Next question, if you look into Nana, next question, arrange the following stages of development of dicot embryo in the order of their occurrence like this. So yes, dicot embryo development, initially what happens ma? Zygote formation, that is first, okay, first zygote formation. Next, globular stage embryo, followed by heart-shaped embryo, and then you can see complete dicot embryo. This heart-shaped embryo you will see only in the embryogeny of dicotyledonous seeds. Heart-shaped embryo you will not see in the embryogeny of monocotyledonous seeds. So here you have to choose the option if you are following the stages accordingly. First is formation of zygote, that is C. Second is formation of globular embryo, that is option 2. Next, A, that is heart-shaped embryo. After that, matured embryo. So completely you have to choose the option 2 beta. Next question, if you look into, arrange the following steps of experiment performed by Griffith. Frederick Griffith has given us what? Transformation principle or transforming principle. Transforming principle by the model organism, that is Streptococcus pneumonia, by taking the bacteria, that is Streptococcus pneumonia. So in Streptococcus pneumonia, if you take two variants are present. One is S strain and the other variant is R strain. Okay, one is S strain and the other variant is R strain. S strain bacteria has shiny coat over the bacteria, which makes the bacteria protective inside the host. Okay, whereas R strain, that is rough strain bacteria, which doesn't have an uncoated layer around the bacteria. So that is R strain. So if you look into considering this S strain and R strain, when they are injected into the rat, different responses are shown. First, what are the consequent steps of this experiment is what asked. First, S strain bacteria is injected into the mice. Mice will die because S strain bacteria has a very strong thick coat over it. Okay, that will protect the bacteria from host environment. Second, if you see S strain, which is heat killed S strain bacteria, when it is injected into the mice is alive. Okay, because the coat is dissolved by the heat. Third, what happened? They have injected R strain into the mice where the mice is alive. Okay, where the mice is alive because bacteria is dead because there is no protein coat around the bacteria. Next, S strain, that is heat killed, and R strain, which is live. S strain heat killed and R strain live. When it is injected into the mice is dead again. Okay, mice is dead. That is where of this final step he has observed that something is being transformed from the heat killed S strain to the R strain, which is leading to the death of the mice. So this is the following consequent steps are in order only. So we have to choose A, B, C and D beta. That is option 1. And next question, if you look into beta, match list 1 and list 2. Metabolic disorder. Okay, metabolic disorder is phenylketonuria, which is also autosomal recessive disease. And pedigree analysis will give you the family tree over the generations. And thalassemia, that is quantitative disease, which is autosomal recessive Mendelian disorder. That is a quantitative disease. Thalassemia is a blood disease beta. That is either due to high quantity of alpha globulins or low quantity of beta globulins. Based on that, we have two types of diseases, that is alpha thalassemia and beta thalassemia. So that is a blood disease. Next, trisomy of 21st chromosome. Just now in the previous question also we have seen trisomy of 21st chromosome is Down's syndrome. So if you clearly look into the options beta, A is option 3, B is option 1, C is 2 and D is 4. So option 3 is the correct answer. Next question if you see. So identify the statements true for RNA. This is from molecular basis or molecular biology, wherein RNA acts as genetic material for some viruses. That is true. Okay, those are retroviruses. RNA also functions as adapter molecule. Of course, that is true. For example, tRNA or soluble RNA, which acts as adapter molecule in the time of translation. RNA has hexose sugar. Wrong, because RNA has pentose sugar. RNA has pentose sugar. Okay, and then RNA also acts as catalyst in some cases. True, RNA acts as catalyst. For example, ribosomal RNA. So here if you look into beta, A, B and D, these three are correct. So in the question they have asked us to choose the correct answer and true statement. So that the answer is A, B and D. Option 2 will be the correct answer. Next question if you look into beta, match list 1 and list 2. So here if you see ribosomes are used in the process of translation. Ribosomes are used in the process of translation, and histone proteins, which are positively charged because of basic amino acids like lysine and arginine, they are used in the nucleosome model, wherein negatively charged DNA wraps around the positively charged histone proteins and thereby DNA condensation occurs. Next, DNA polymerase. Okay, DNA polymerase that is used in the process of replication. RNA polymerase is used to synthesize mRNA during the process of translation. So if you clearly look into the options beta, A is 3, B is 4, C is 1 and D is 2. Option 4 will be the correct answer. Next question if you look into beta, in DNA, N-glycosidic linkage is present in between. N-glycosidic linkage is present in between. Just let us quickly look into the nucleotide ones. Okay, let us quickly look into the nucleotide ones. So C1 of this pentose sugar is attached to the nitrogen base by what linkage? Glycosidic linkage. Glycosidic linkage. Okay, and carbon 2, this is 3, 4 and 5th carbon. Okay, 5th carbon is attached to phosphate group by phosphoester bond. By phosphoester bond. Okay, so for the formation of one nucleotide we need one N-glycosidic bond and one phosphoester bond. Here they have asked us in DNA N-glycosidic linkage is present in between nitrogenous base and pentose sugar. So this is option 2. Okay, that is option 2. Next question if you look into which of the following crop was made resistant to yellow mosaic virus and powdery mildew? This question is previously also asked in one more paper. That answer is Mung B, which is directly given from the strategies of crop production. So that chapter is very important. This is produced due to mutational breeding and this variant is resistant to yellow mosaic virus and powdery mildew. And next questions if you look into beta, match list 1 and list 2. Lymphoid organ. Lymphoid organ is related to what? Bone marrow and thymus. Okay. Cancer is caused due to carcinogens. HIV is detected through ELISA and typhoid is detected through Widal test. So here if you clearly look into options, first A is 4, B is A is 3, B is 4 and C is 1, D is 2. Option 2 is the correct answer. Next, this is also from tissue culture. Okay, this is from tissue culture, wherein sterilized plant part you call it as explant. Sterilized plant part you will call it as explant. Genetically similar plants are called as soma clones. Genetically similar plants are called as soma clones. Meristem. Meristem is nothing but virus free culture. Meristem, virus free culture. Next, somatic hybrids. The best hybrid that is given is pomato. That is a cross between somatic embryos of potato and tomato, and the hybrid we will get is pomato. So here if you look into options beta, A is 3. So yes, sterilized A is 4, which is explant, and genetically similar plants, that is 3 soma clones. Meristem, that is 2 virus free culture. Next D, that is somatic hybrids, that is pomato, option 1. So here option 4 is the correct answer. And next if you look into the varieties beta, this question is also asked from strategies of crop production, wherein Sonalika and Ratna respectively are high yielding varieties. Sonalika and Ratna are high yielding varieties of what? Sonalika is the variety of wheat and Ratna is the variety of rice. Okay. Next, which of the following is not a nitrogen fixing microorganism? You have to clearly look into it because two options are very similar. First is Azotobacter, Oscillatoria and Nostoc. These are nitrogen fixing bacteria. Acetobacter aceti, this will synthesize or it will give out acetic acid. Acetobacter aceti will give acetic acid. This we have learnt in the microbes in human welfare chapter. Whereas these three, Acetobacter, Oscillatoria and Nostoc, they are nitrogen fixing microorganisms. So here the option will be 3. Okay, when the options are in close similar, you have to look into it keenly. Just by taking one more minute, if you look into it keenly, you can answer it. Next question, ma. The large holes in the Swiss cheese. The large holes in the Swiss cheese are produced due to production of large amount of carbon dioxide. That is produced because of large amount of carbon dioxide that is released due to alcoholic fermentation. Due to alcoholic fermentation. Fine. So at the end of alcoholic fermentation, what are the products that are released beta? The products that are released are ethanol and carbon dioxide. Fine. So these are being released. Next, which of the following is not a cyanobacteria? Here you can see Nostoc, Anabaena and Oscillatoria. All these three are blue-green algae or cyanobacteria. When it comes to glomus, glomus is a fungi. Glomus is a fungi. So here that is option 2. Next, agarose, a gel used as matrix in gel electrophoresis, is a natural polymer which is extracted from. This is extracted from seaweeds like. These are extracted from seaweeds like Gracilaria and Gelidium. Fine. From this red algae which are seaweeds, agarose gel is extracted, that is used in gel electrophoresis. Next, the first instance of construction of artificial DNA molecule was carried out on the plasmid of, that is first carried by the plasmid of Salmonella typhimurium, that is option 4. Okay, that is option 4. Next, antibiotic resistance gene. Okay, antibiotic resistance gene in a vector helps in the selection of, first primarily it will help in the selection of transformed cells. If in case in your question transformed cells option is not given, then you can also go for recombinant cells, but primarily it will help in the selection of transformed cells. Later you can also go through recombinant cells, but here in this question we have to choose only one option, right? So the most accurate answer will be transformed cells. Next, identify the gene which is not effective against cotton bollworms. which is not effective against cotton bollworms. Cotton bollworms are majorly identified by Cry1 AC and Cry2 AB. Okay, Cry1 AC and Cry2 AB. So Cry1 AC and Cry2 AB, they are against the cotton bollworms. Cry1 AC and Cry2 AB, they are against cotton bollworms. Cry1 AB is against corn borer. Okay, corn borer. Keep that in your mind, because many at times the question is asked with respect to this. So identify the gene which is not effective against cotton bollworms, that is Cry1 AB, because that is against corn borer. Next, the process RNA interference was used in tobacco plant to develop resistance against RNA interference. Most important concept. This is recently asked in NEET also. For 2 questions RNAi is asked. Okay, so here the process of RNA interference used in tobacco plant to develop resistance against nematodes. This is to develop resistance against nematodes. Next, common term used to refer the use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment to countries and people. This is related to biopiracy. This is related to what, ma? Biopiracy. All these terms we have to be aware of, that is bioinformatics, biopiracy, biopatenting and biological theft. These terms we have to have an idea about the meanings of those terms, okay, because they will be asked usually in the CUET entrances. Okay, and last question of this session, out of the following plants, which one is not browsed by cattle? Cattle, this you can easily answer with general knowledge also. It will usually browse wheat, cowpea and sugarcane, but not Calotropis. Calotropis is not browsed by cattle or herbivores. Okay, so with this we can end the previous year question sessions of CUET. In coming up sessions we will also do CUET concept sessions in very short, which is very useful for all the students of BPC 11th 12th standard and who are going to appear for CUET 2026 exam. I would like to end the session by giving you a grand announcement that here in Narayanguda, Hyderabad at NEET World, CUET UG entrance program is getting started from May 20th and second batch is from June 1st. You can also take the course on online. To know about the information, you can call on to the below number. Thank you.